Bom dia a todos. Vamos começar essa sessão. So, first of all, perhaps you don't know me. My name is George Khalil. I am the professor of clinical immunology and allergy, head of division of this specialty at the School of Medicine, University of São Paulo. And I'm very glad that, again, we have this course here. It's one of the disciplines that are one of the best disciplines, I would say, of our doctoral program that in Brazil we call postdoctoral program, actually. And Esper has been very active in this program, and this course has been very important. And I'm doing this introduction to let you know that 12 years ago, I invited a young investigator that was doing a very beautiful career, I would say, to join our group. And this was Esper, 12 years ago. And he came as an assistant professor. And here at the University of São Paulo, in order to become an associate professor, you have to do a very hard exam that doesn't exist in United States or England, but it does exist in Germany. That's a docent. That's a different exams that we do. It's very tiring. It takes 5 days, and after this, he became associate professor. And in a few weeks ago, I would say, a few weeks or months already, months ago, after a very hard, also open concourse, I mean, to see who would be the professor, he was nominated as full professor of infectious diseases. So he's leaving immunology, he's going to the other side, to the microbes, but still looking for both sides. So he is now one of the full professors of infectious diseases at the same School of Medicine. And we are of course, very proud that he got this position. At the same time, we are a little bit sad because he's not only dedicated to us, but we are pretty sure that he'll continue something that's going very well, that the work that we're doing, because since Esper came those 12 years ago, has done many interesting things in our group. He brought many new technologies, new ways of thinking, and also many international researchers that we have here this time. So we have people from all over that come for this course that I think has been more and more important in the area. And I think that the people that come from this course, they come because they know that they are helping you in understanding HIV, but they also are coming because they want to know what's going on with their colleagues, because it's a pretty informal way of knowing what's going on. It's different from a formal meeting or formal meeting at the NIAID to see how the RO1s are going. And so this is an informal way of networking. And this is something that you should also take advantage of, network with these people, very important people, very hard to have an appointment if you go to their places, but they are here and they are dedicated to teaching. And I'd like to thank all the speakers that came again this year. I'm living in Boston those days, and I came also to join the group because I think it would be important. And I'd like to thank again Esper for being so helpful to our discipline and contributed so much to clinical immunology and allergy. And I really hope that you'll continue helping us working in different areas of mutual interest. So thank you, Esper. Now I'd like to introduce a very good friend, Barney Graham. Barney Graham is a physician scientist or a scientist physician. I don't know.
It switches.
It switches. But it's someone that's very interesting because he does very fundamental work. At the same time, he has a medical view. So he is involved in developing new molecules, but at the same time, he's looking for all the development of the vaccines till the end for clinical trials. And I don't know if you have seen the news. A few weeks ago, they started the phase 1 clinical trial of a new universal vaccine against influenza. That's what everybody's looking for, for many, many years. Because, every time that you have influenza, seasonal influenza, you have to get vaccinated because the serotypes vary. Well, if you have a universal vaccine, this would not be the case. It's a tremendous work for global health, and they just started. And being a deputy director of VRC, that's the Vaccine Research Center in NIH, a beautiful building, beautiful people, and very collaborative. We have many people here, like Rick and Adrian as well. He has been working in many different diseases, infectious diseases that are very important for mankind. Diseases, so he worked on HIV, but he has been working on flavivirus, dengue, Zika. We are testing a Zika vaccine that is being developed in NIH. And he also worked on respiratory syncytial virus. So diseases that are very important for global health. And he has a very interesting view because, as I mentioned to you, he looks at the very basic research till the end for production, and he's involved even in the different steps to get it produced, sometimes at NIH, sometimes he has to order it for being manufactured elsewhere. So, Barney, we are very happy and very proud to have you here, very honored to have you again. And
thank you, George.
Looking for your talk.
Thank you very much. And it's my pleasure to be here. I come here largely because of George and Esper, and because of the other faculty that are here. I probably learn more in this meeting than I do all the rest of the meetings of the year combined. So you are very fortunate to have the faculty that you have here. And I'll just let you know that I started my timer with his introduction, so my stopwatch will stop me on the same time, even though it was too long. All right. Okay, so first, let me just introduce briefly the VRC. The Vaccine Research Center at NIH started around 19 years ago. And we now have these 13 principal investigators and program directors that lead the group that works in this building 40 on the NIH campus that you're welcome to visit if you're in Bethesda. And we were founded and started to work on HIV vaccines. It's still our major purpose, and I think you'll hear more from Adrian McDermott and Richard Kaup, who's the other deputy director at VRC, on Monday about some of our work on HIV and AIDS. But Esper gave me the opportunity today to talk about some of the things that have derived from our work on HIV to work that have worked on simpler viruses. So the work on HIV has really led to the ability to do a lot of other types of interventions on viruses that are a bit simpler to solve than HIV. And so now we work on all these different programs from A to Z. We make vaccines based on several different platforms, gene-based, virus-like particle, protein-based approaches, or subunit particles, or proteins displayed on nanoparticles. Dr. Khalil just told you a little bit about our flu program. This headless stem trimer just started in trials last week. And we also make a lot of monoclonal antibodies that are being used for HIV prevention that I think you'll hear about from Dr. Kaup, and that are being used for treatment of Ebola patients in East Congo now. Our program is fortunate and privileged because it's not only basic research, but we have process development, people that are chemical engineers and mechanical engineers who help us turn research-grade products into actual products that can be made by good manufacturing practice at our pilot plant in Frederick. put it in a vial. We have a big clinical group who can test these in phase 1, and then a group also in Gaithersburg led by Dr. McDermott who does the analysis of our clinical trial samples. And so we have this capacity to at least take things through phase 1 trials. A few disclosures, and what I'll do today is talk about emerging viral diseases, talk about some of the technologies we now have to make better vaccines and some of the technologies that can help us make vaccines faster. So we know, I don't have to explain this, we know we're having more and more emerging infectious diseases. Just in the last few years, we've had MERS coronavirus, chikungunya come through the Americas, bigger outbreaks of Ebola, and more recently Zika. And so we are going to be pestered by these emerging infections, especially virological and virus infections that continue to come through both zoonotic and vector-borne pathways. Zoonotic meaning from an animal. About 75% of the new virus infections that we emerge come from animal sources. The others come from vector-borne sources like Zika. And traditionally what we've done is to quarantine, passive surveillance, contact tracing. We try to use what's available already, but it's like the little Dutch boy with his finger in the dike and it's barely holding together. And about 40 years ago now, we started seeing that the dike is leaking in so many places. It's no longer possible to just have these simple approaches. So in '76, Ebola emerged in Congo. We were threatened by what they called swine flu. Legionnaire's disease came. And since then, we've had a number of zoonotic diseases like HIV and MERS and SARS, the coronaviruses, and vector-borne diseases. And we're being re-threatened by diseases like yellow fever and flu and measles and polio that keep coming because our traditional public health approaches are not sufficient. And for these, even though we have some solutions, we can't manage those, and we can't prepare for all these other viruses that have the potential for pandemic display. So we need a new system. And so what I'm going to try to give you is a different way of thinking about how to prepare for the future. We need better surveillance and discovery. We need to be farther ahead in a proactive way on doing the basic virology and solving structures and things like that. And that would allow us to do antigen design, which is what I'm going to talk about today, and then platform manufacturing approaches that will allow us to be faster. So I really like this study, which was simply a person sitting down with the literature and asking what were all the viruses that came in the 20th century. And so he just simply made a list of all the viruses that infected humans during that 100 years of the 20th century, and then asked, there obviously linear increase in the number of viruses, but what he showed was that there was a plateauing effect on the number of viral families. So there's around 110 virus families, but only around 23 to 30, depending on how you count. The Bunya viralis has now become an order, so it added number of families. But the point is the number of virus families in humans that infect humans is plateaued, and it's around a couple of dozen. And so it's a finite number. It's not an infinite number. So what can we do to address the problem of emerging infections? And even though they're going to keep coming, we now have a number of new technologies that have come, especially over the last 10 years, that give us new tools that can be used, like structural biology, which I'll talk about today, protein design. I think Adrian McDermott on Monday will talk more about this idea of rapid isolation of human monoclonals and what we can do with sequencing to identify lineages of antibodies that may be more productive. And so the idea here is that most of our vaccine developments in the past have been opportunistic. These first 4 from Jenner through Pasteur and coming up to influenza were maybe more persistence by a single individual and very just based on empiricism. But this cluster of vaccines was based on the ability to do cell culture and grow viruses in vitro. It allowed the development of polio, measles, mumps, rubella vaccines. This cluster of vaccines since the 1980s is really based on our understanding of molecular biology. Allows you to make molecular clones and understand reassortment and make recombinant proteins and virus-like particles, et cetera. And so the question is whether any of these new technologies can we take advantage of and opportunistically develop new vaccines for some of the remaining problems and for some of the future problems. So especially structural biology, this ability to do single-cell analysis, sequencing of immunoglobulins from individually sorted B cells, and the high-throughput sequencing. This cluster of technologies has really transformed the way we think about making vaccines over the last 10 years. So to prepare for these new viruses that are likely to come, there's a few different ways people are approaching these, and Dr. Fauci outlined these last year, a year or two ago, and they include a prototype pathogen approach, which I'll describe today, the priority pathogen approach, which WHO and CEPI and places like that have taken, where they just say, CEPI said MERS, coronavirus, and Lassa and Nipah virus were their top priorities, and let's just work on those three. But if there's more coming, just those three are not going to help us very much. And then the platform approach means that you use a manufacturing technology that can be consistent between products and allow you to more quickly get through the regulatory process and more quickly make products that used to take several years. So I'll start with a story about respiratory syncytial virus. It's a virus, it's a pneumovirus. It used to be a paramyxovirus. It's now a pneumovirus. It's an orthopneumovirus in the pneumovirus family. It infects babies. It causes severe disease in infants, about 120,000 deaths per year, and more than 3 million hospitalizations per year occur. And it infects everyone. It infects everyone repeatedly throughout life. So young infants, school-aged children, young adults. It's particularly serious infection in bone marrow transplantation. And then it gets us later when we become more frail and elderly. And so it's a big cause of mortality and morbidity in both the elderly and the very young infant. It's also associated with the induction and of asthma or wheezing, and I'll show you why we think that is. This is the gene order of the virus. We're mostly going to talk about the surface proteins, which is the target of vaccine development, especially the F-glycoprotein, which is that mediates entry. Briefly, just to show you why this airway damage and wheezing occurs, RSV infects the ciliated bronchiolar epithelium in the small airways. And you see it's almost circumferential infection when it occurs. It spares the basal epithelium, so it can be regenerated. And then it also infects the type 1 pneumocytes in the alveolus. And what happens in these young infants is that between the inflammatory debris and the sloughed epithelium and the fibrin and then the mucus, they get airway obstruction because their airways are so small. And that airway obstruction is what leads to the severe disease and hospitalization, but your lungs are also developing during those first 6 months of life, and so airways even are still dividing. And when children get this kind of a serious infection early in life, it can affect their lifelong airway physiology. So how do you attack RSV? This is a virus budding from the cell, and I'm showing you the cryo-EM of the membrane. And you can see both short and long forms of this F protein on the surface. This is the short and long form, and they conform perfectly to the structures of the prefusion and the postfusion rearranged molecule. So on RSV viruses, the fusion protein flips spontaneously into its inactive form. And I'll show you why that's important. RSV is a class 1 fusion glycoprotein. It's very much like the class 1 fusion protein of HIV. It even shares some sequence homology in its fusion peptide. And so understanding how these fusion proteins work and how the structural domains are somewhat different, but the function is all the same. And that is that there's a fusion machine that has heptad repeats near the fusion peptide and the transmembrane domain that rearrange and fold together and pull the membranes together to allow a fusion pore to be established and then the virus to enter the cell. And so RSV is shown here as a pre-fusion form of of the F protein, similar to the paramyxovirus, and now people are using Lassa and Ebola and the flu and coronaviruses and solving these structures of the functional class 1 fusion protein. And here's an example of how these proteins work. For instance, RSV, it's triggered by something on the membrane to release the fusion peptide that penetrates the host cell. These heptad repeats fold together, they're attracted to each other, and it becomes this anti-parallel 6-helix bundle that creates the fusion pore for entry of the nucleocapsid. So you start with this protein and you end with this protein. The reason this is important is because over the last 50 years, there's been at least now 7 clinical trials using this form of the F protein because it wasn't really understood that this existed. And so this rearranged version of F has failed in 7 trials going from the early 90s to just recently. And so what you should see here is that after this structure was solved, you can see that the base of this molecule has sequence homology or structural homology to this part of the post-fusion molecule, but the top of this and this part of the molecules are unique. And so the post-fusion was used because some of these conserved structures were immunogenic and could elicit neutralizing antibodies. The problem is those antibodies were not that potent. So even the palivizumab antibody that's used to treat premature babies and is licensed for use in that way has potency up in this range. And the antibodies that see the top of that prefusion molecule have potencies that are 100 to 1,000 times higher than you would see for even the palivizumab. And the post-fusion specific antibodies have very poor neutralizing capacity. So these 2 sites, these 2 epitopes or domains on the top account for more than 70% of the high potency antibodies that you can induce in humans. And so that's what we want to target if we're making a vaccine. Another way of thinking about this is that these 2 major epitopes that were preserved on the post-fusion structure are also present on the pre-fusion structure, or otherwise they wouldn't be able to neutralize. But these other antibodies that recognize the top and this one on the side are the ones that really have the high potency. And then this one that binds mostly the post has no neutralizing capacity at all and can actually interfere with neutralization by some of these other antibodies. So finding that prefusion flips to postfusion and understanding the structure of this, mapping the epitopes, and finding that the high neutralization sensitive epitopes are present only on this form led to the stabilization of this molecule. A trimerization domain was put at the C-terminus and some stabilizing mutations were put inside, and now it's a clinical product. We just finished the analysis of this early phase of the clinical trial where we did dose escalation from 50 to 150 to 500 with or without alum, and I'll show you some data from these first 40 subjects. So this is the main story, that at either 50 or 150, with or without alum, you were getting between 12 and 15-fold increase in neutralizing activity with a single injection of this protein. In the past, these other 7 trials that have failed have been inducing about a 2-fold increase in neutralizing activity. They can induce 30-fold rise in binding titers, but they only induce 2-fold in neutralizing titers. So this new molecule is able to access the precursors that have high neutralizing potential, and so we're excited or hopeful that this may make a much better vaccine. You can prove that these antibodies that are elicited are prefusion specific because here's the neutralization response at 0, 4, and 12 weeks after this injection. And here is that response when you adsorb out all the antibodies that bind the postfusion. So removing those antibodies to the shared surface still preserves this neutralization response, but if you adsorb with the prefusion molecule, you lose everything. So these antibodies that are being made at either dose are highly prefusion specific. And you can show that also by using these specialized probes that the VRC has evolved over time for its work on either HIV or flu. And using these probes of either the pre or the post-fusion form, you can show that most of the B cells responding to this vaccine are in the prefusion specific category, and some of them are dual binding, binding both the conserved epitopes on the post-fusion. You can use these probes to then start understanding not only the magnitude of the antibody response, but you can start looking at memory B cell responses and show that you can induce memory B cells with IgG and other, and actually change the threshold or plateau of where these memory B cells have been established. This never happens if you get reinfected. So this vaccine is working better. It's a supernormal neutralizing activity, and it's also changing memory B cell populations better than an actual infection would. It's also inducing IgA, which is interesting because these are all people who've been repeatedly primed mucosally, and now with a parenteral protein, you're able to draw these memory B cells out into the blood that are making IgA. And you're also eliciting serum IgA responses, and we haven't measured mucosal responses yet, but we're hopeful that this translates into also boosting some of the mucosal responses. So the point of this is that, first point is that basic research can sometimes be profitable, and that solving the atomic structure of F revealed this new target of vulnerability, and then it was a chance to prove that structure-based design and protein engineering at the atomic level can really improve or result in a better vaccine antigen. And for these reasons, which I just explained, and it also has given us these reagents for the pre and the post, have also given us reagents that allow us to now do more detailed phenotyping of the B cell memory, learning maybe how to make longer, durable B cell responses, and also allow us to probe the repertoire in much more depth than has ever been done for RSV before. So how would this be relevant to pandemic preparedness? Well, I showed you that RSV is just one of many class 1 fusion proteins, and so one of the questions is, can you learn and generalize the lessons from RSV to other viruses that are enveloped and have class 1 fusion proteins? So I'm just showing you some of the known structures of prefusion molecules here, and I'll tell you 2 brief stories, one from coronavirus and one from another paramyxovirus, in which being able to solve the prefusion structure of the spike protein of coronavirus allowed us to identify places where you could do stabilization. So just 2 proline residues at the top of this central helix, which is part of that fusion machine that I was explaining in RSV, largely stabilizes this viral protein. And we did this with an endemic coronavirus named HKU1. But when we found these 2 proline residues could stabilize, we applied those to the MERS and SARS coronavirus structures, and replacing those same 2 residues with proline finally allowed us to identify the prefusion structure of the MERS and SARS coronavirus. It also improved their expression, and these molecules were too unstable before this stabilizing to ever find their exact structures. But it's also now allowed stabilization and identification of structures for more than a dozen different coronaviruses. And so this information being added to the database now allows you to think about generalizable approaches to stabilizing spike proteins if we happen to have a new coronavirus outbreak with something like this WIV1, which is a bat coronavirus, but it's very SARS-like. And it also allowed us to use the MERS coronavirus spike as a reagent to find new antibodies. So in the past, the only antibodies ever really present for MERS coronavirus was to this receptor binding domain that's shown here, overlapping the adjacent protomer. But now we have antibodies to the stem region of MERS and to this N-terminal domain of MERS that can affect receptor binding domain access. So having these reagents ahead of time is going to significantly improve our ability to respond to a new coronavirus outbreak if it happens. We can also show that these prefusion spikes are more immunogenic or more protective than the post-fusion form of the molecule or a monomeric form of the molecule. And just in this mouse experiment, you can go down to 0.1 or even 0.01 micrograms and still show immunogenicity of this prefusion molecule. And using that, what we call the S2P, the red bars, we can generate broad neutralizing activity against many coronavirus strains and also at a higher level. And in DPP4 in knock-in mice that can grow the SARS or the MERS coronavirus, you can show that a very low dose of this immunogen can fully protect against lethality and weight loss. So this could be a generalizable approach for coronavirus proteins, and this is what I've just told you. I'm not going to go over that again. So what about Nipah? Nipah is a paramyxovirus. The structure was already known, but there was virtually no monoclonal antibodies or anything to guide stabilization of this structure. And so we just took the structure and looked and tried to define whether we could see a disulfide or a cavity filling or other approaches we've learned in coronavirus to stabilize the molecule. And we're guided by expression levels. So just simply adding a few stabilizing mutations also improves expression from cell culture, which was shown here. And many of those designs ended up giving you a prefusion stabilized F protein. And we've now made those F proteins. We've even done some interesting, innovative designs. Guillaume Stewart-Jones and John Mascola's lab has put G proteins on the base of the tip of this prefusion molecule, and now we have a FG chimera that's being tested in ferrets to see if it can be protective. So as we try to get back to HIV, you know, HIV, as you'll probably hear in other talks, is a very difficult virus to neutralize because its glycoprotein has so many ways of evading neutralizing activity. The genetic diversity is one of the biggest. It's very heavily covered in glycans that prevent access to the neutralizing sensitive sites. It has conformational evasion, and it has problems with immunodominance. And so this idea of stabilizing the prefusion form was really, I think, all we needed for RSV. So it turned out to be a fairly simple problem. Influenza presents problems because it's genetically diverse and it has immunodominance and redirects the immune response to places we don't want. We're starting to solve some of those problems, and then the coronavirus spike turns out to be heavily glycosylated. So the question is whether we can take the learning that is coming from these other viruses, made possible by the technologies that were developed working on HIV, and then reapply them back to HIV and end up with better solutions. And some of that has already started. This is a stabilized envelope structure that comes out of Peter Kwong's lab at the VRC. And so the conformational evasion problem of HIV envelope has now been solved. The question is, can we solve all those other problems and make a more immunogenic envelope protein for HIV? Okay, so how do we prepare for the next pandemic? I'm not going to say anything about influenza at this talk, George, so thank you for that introduction. It's all going to be other things. So how long does it take to make a vaccine for other viral diseases? I've listed some of the times here, and it's important. The most important thing that's happening in this room today is we're speaking to young people. And young people are needed to develop vaccines because you need to give yourself at least 3 decades if you want to have a chance to do this. It takes a long time to develop vaccines, even with these new approaches. And so you can see here, from the time of virus discovery to the time of vaccine licensure has been decades. And for flu, you could argue that maybe there was a vaccine after 12 years, but it's really an effective vaccine has now taken more than 84 years. For RSV, it was discovered in 1956. We're now over 60 years. HIV and HCV, we're still waiting, but all these other vaccines like HPV, which people think is an effective vaccine and maybe a simple vaccine, still took 3 decades from the time of discovery to the time of vaccine availability. So we need you to start on vaccinology at your age so you can finish before you retire. Some of the tools you have are better than the tools we had, and one way to think about that is this idea of synthetic vaccinology. You know, it used to be that you found the virus, you learned how to grow the virus, then you either inactivated the virus or attenuated the virus, and that was your vaccine. Most of our licensed vaccines, all but 2, are based on whole viruses. You have to learn how to grow the virus and activate the virus. It takes a very long time to get it just right. Now, you can argue that you don't really even need the virus anymore. If you made a diagnosis of a new virus on the other side of the world and could sequence even some of the genome, you could send that data electronically over the internet, and within 2 to 3 weeks you could have constructs made. You could start making your own proteins. You could use those proteins to generate new monoclonal antibodies. You could start designing vaccines based on how we know how to design antigens now. And so you could start a vaccine program without ever having the virus, and you could have reagents and be going within 3 to 4 weeks. It's a very different world. An example of that is the Zika vaccine development, where Zika emerged, as you well know. It was largely happening here in Brazil. And in this case, because of the VRC experience with DNA vaccine manufacturing that had gone back into the early 2000s, we were able to very quickly identify sequences that had worked for making a West Nile vaccine 10 years earlier. And within 100 days of selecting that sequence, we were able to start a phase 1 trial with a candidate DNA vaccine, which then was in phase 2 testing within about a year. This was an unprecedented speed of going from nothing to a phase 2, but unfortunately it still wasn't quite fast enough. So this idea of platform technologies where you are very familiar with a way of expressing a vaccine antigen. We did this with SARS coronavirus in 2003, and then the avian influenza scare in 2005 and 6, the recent pandemic flu, the California 09 in 2009, and then recently Zika. And we may be about as fast as we can get at this point, but the idea is that DNA is DNA. And so whether you're making a SARS protein or an influenza protein or a Zika virus protein, the manufacturing approach is the same, and so you can speed that through some of the regulatory process. That DNA vaccine was immunogenic in humans, and it achieved a neutralizing activity at the level that protected macaques from viremia. And so that's what allowed it to go into the phase 2 testing, some of which is being done here in Dr. Callas's clinic right here in Sao Paulo. So that phase 2 trial incorporated sites from all these different places. So there's 17 sites in 8 different countries, including this one in Sao Paulo. So that trial has now been enrolled, and we're waiting to see if we can achieve enough endpoints to get an answer. Although Zika, as you know, is not as common as it used to be. We're hoping that maybe through just DNA and diagnosis of asymptomatic infections, we may still be able to get an answer. So then, how do we look at the bigger problem of what could come at us? So these are potential viral pathogens. These are the families of viruses that are known to infect humans, and we have some licensed vaccines against these, although almost all of them are based on live viruses, and that wouldn't be fast enough to respond to a new outbreak because it takes so long to learn how to grow the virus effectively and safely. We have a lot of work on these kinds of virus families, but very little work on these, and we really don't have licensed products here to guide anything that we do. But what if we took prototypic viruses from this and defined all these properties and at least got vaccines into phase 1 testing? And then the viruses behind those in the families that we know have infected humans, it seems like an infinite number, but it's really only about 120. So there's about 120 viruses from all these 23 families that we should be somewhat afraid of. And the question is, can we do something about that? So we have about 25 we have 25 licensed products in these 13 families and about 17 prototypic viruses in these other 10 families that we need to address, and then about another 120 behind those that we need to understand. So one of the proposals now at NIAID, where I work, is that we do a prototype pathogen preparedness campaign where we learn how to make the vaccines, design the antigens, make monoclonal antibodies, identify all the structures, even of polymerase so we can find therapeutics, and really have a plan and a possible candidate vaccine for at least 30 of these prototype viruses covering all those virus families, and in some cases the virus genuses. And then we could get it through phase 1 and maintain supplies on the shelf ready to go if there's an outbreak so that we can get in faster than we did for Zika, faster than we did for Ebola, and actually get an answer about efficacy during the initial outbreak that can guide the development of other more permanent vaccines and help us understand correlates of immunity that might actually cut across other virus families. And then for all those other viruses, we could generate the information and at least develop candidate approaches and reagents and take them through animal models so that if we had platform technologies, we would know where to go with that kind of rapid response. And so this is a program being proposed at NIAID exploiting these new technologies of single-cell analysis and cloning out of new monoclonal antibodies, structural biology, its ability to rapidly make new animal models with CRISPR-type technology. Again, the single-cell analysis of immune response and all these different ways of designing antigens and displaying them on nanoparticles and delivering them either through protein vectors or nucleic acid itself. And And you can consider dividing it into these kinds of buckets. So I told you about class 1 fusion proteins. There's also class 2 that represent these virus families, class 3, and some other that are unclassified, and then non-envelope viruses. And if we had expert lab and pathogenesis work on all these different categories supported by these core functions that could help turn this research into development activities, then we would have a better way to approach being more prepared for the next pandemic. So vaccines in the 21st century, we need these as public health tools for emerging infections. We have a new era, we're in a new era of vaccinology because of these new technologies that have been developed, largely driven by HIV work. See, that's my timer. It's my last slide. Okay, but we need advanced preparation. So we need better surveillance. This is not what our group is skilled at, but we need surveillance and discovery efforts around the globe to fill in what I call the periodic table of viruses, so we'll know what's coming at us and know how to be prepared. We can do this part, the basic virology and prototype antigen designs, but we need more engagement of industry to really develop these platform technologies to a greater extent. We also need new business plans because many of these problems are public health problems and not really commercially viable things to work on. And so we need more options for government to government or public-private partnerships so that we can address the problems that have public health value but not commercial value. And then we just need to all get on the same page more quickly when these crises emerge. So I'm going to just stop and thank my laboratory that's pictured here. You heard about the work on the flu. This man, Masaru Kanakiyo, is largely leading that effort on the influenza. Dr. Ruckward is leading the work on my lab on respiratory syncytial virus. And Dr. Morabito has led a lot of the work that we did on Zika. And Dr. Corbett here is leading the work on coronaviruses. I want to thank the people I work with at the Vaccine Research Center, especially Dr. Mascola and Peter Kwong's group, along with this person who's now an associate professor but was a postdoc at the time we started that work on respiratory syncytial virus in Peter Kwong's lab, and our collaborators in Xiamen, China, who were instrumental in helping us find those prefusion-specific antibodies, and all the other people in NIAID, and then our clinical group led by Dr. Ledgerwood, sitting here, who has generated all this clinical information that helps drive our programs. So thank you very much.
Well, thank you, Barney, for this very comprehensive overview of vaccine production and field results. If you want to sit here, you'll be more comfortable.
Okay.
So do you have questions from the floor?
Barney, this is an enormous effort, and you know, I can see downstream great value, but what do you do, what can you do to prevent vaccine denialism, which is an issue for so many of the vaccines that have been established and work?
So an anti-vaccine question. Well, you know, we've been
I'm
working
not saying
on,
it because I like it. I'm just...
We've been working on HIV vaccines since the mid-'80s. And so a lot of what I would say is related to that early work on HIV vaccines. And at a time before we even had treatment for HIV, we were trying to convince a healthy person who was otherwise healthy and uninfected to take an injection of something that would make them HIV positive. And, you know, would a healthy person volunteer for an HIV vaccine trial? That sounds pretty scary. And would you advise your daughter to do something like that, for instance? So, you know, this fear of vaccines, especially experimental vaccines, is deeply rooted in beliefs. And, you know, a lot of times what we take to people are facts. And facts and data are not as interesting as belief and hope. You know, and people who use herbal remedies for different things, you know, hoping that they'll work and being convinced and believing that they'll work is a lot more attractive than taking tetracycline that, you know, has a certain percentage of side effects and a certain percentage of failures, and it's so dry. The data we have to support tetracycline use is, you know, it's not very compelling. So I think what we have to do as doctors and scientists is to convince people, to try to teach people how vaccines work. If they really knew more about the details or basics of just how a vaccine works, take some of the mystery out of it, it give them a new thing to believe in. Believe in immunology instead of believing in something else off the internet. And so I think we have to work at a different level and not just present facts, but try to educate people about how vaccines work. And I don't know any other way to do it. I mean, you get some traction during crises like the measles outbreak, but when we have measles outbreaks, people go back and take a measles vaccine. And when we had a smallpox scare back in the 70s, people lined up for city blocks, around city blocks in New York to get their vaccine immunizations. But, you know, unless people are scared, they won't do that. And so on a routine basis, we have to just get people better educated and have a way to believe in vaccines that's different than just based on facts.
I would like to comment on this. We are scared in this country, in Brazil. We had one of the best programs of immunization, vaccination. It covered about 98% of the target population. It dropped in the last 3 years to 70% coverage. And so where the facts come from, there's nothing really. I was a little bit worried when I was teaching in the doctoral degree, and I asked for the girls or men, young men that were in the room if they were vaccinating their kids, and they said, we are trying to avoid, just find a way of avoiding. I mean, you're immunologists, I mean, you're here. How can you avoid it? She said, well, you know, I have never seen measles. Measles, it doesn't happen anymore. Some people claim there are problems. I don't know, but since I have never seen measles, I avoid to vaccinate. That's terrible. And we are really discussing at the government level. There is a committee of scientists that kind of counsel the immunization program. What should we do? And we cannot control what they call the social media, and people can publicize whatever they have in their minds, and they don't have to prove anything. There is no responsibility, and this is doing a great damage in this country.
I think we have to be careful not to attack the beliefs of the anti-vaxxers. We need to replace their beliefs. We shouldn't attack their beliefs. It needs to be non-judgmental. Otherwise, the two groups just get further and further apart.
Esther?
Yeah. How do you see we can make this a global effort, Barney? Because you don't know where the next pathogen will emerge. And having more engaged countries in providing the necessary infrastructure and preparedness to come up with a solution.
Well, first, I think NIAID has to get its act together and be more organized, because a lot of this stuff happens, but it happens on a very random, non-systematic way. And, 100 years from now, maybe we'll figure this out. But to me, the first step has to be a better global surveillance network. So we need to have sequencing, high-capacity sequencing activity at all the places around the equator. So most emerging infections are coming out of tropical areas. And I like to think that most pandemic threats begin as regional problems. And so if you could really have better surveillance, better virus discovery, and build technology that not only does surveillance, but discovery for research, and then you use the same technologies for doing diagnostics and clinical support, you could justify building out that capacity for a number of different reasons. And I think if you had better surveillance and build out that periodic table of viruses, that you would just be able to maybe work earlier, even with traditional techniques, and keep things more regional. I mean, can you imagine what would have happened if HIV could have been diagnosed in Uganda or Cameroon in 1960s? It would have been a regional disease. And so I think you have to first get that set up, and then there needs to be clinical infrastructure everywhere if we want to be able to quickly respond to outbreaks. Instead of having to build, I think Dr. Ledgerwood's team interviewed 158 groups and sites to find 17 sites that could do this Zika work, and it took an enormous effort. And so if you had established infrastructure and capable clinical sites around the world, that would be another big start.
Barney, well, I completely agree with you, but we need a better vision of the people, our politicians, right? Or the decision-making people. I can see around the world, not only in this country, we're facing many problems in science and how to provide this type of information, but I can see the struggle around the world if I take, for instance, ICGB, that's the International United Nations Institute, where they're trying to keep the world working in developing countries or centers like Senegal, the Pasteur in Senegal, where they have a very nice work that they did for Ebola and for yellow fever and so on. I mean, they don't have the money because sometimes this doesn't bring votes to be in preparedness. In the States, yes, but not in many of those countries, unfortunately.
So somehow we have to do a better job. Companies can't stay in business by making these things unless they're paid in dollars, in actual real dollars. And the problem for many of the diseases, the value is in public health. And so those public health value doesn't pay a company in dollars, and so the companies won't do the work. So we have to have government entities or nonprofit or philanthropic organizations that can actually do the work based on public health value. We haven't done a very good job of translating public health value into dollars. It's estimated that if we had a 1918 flu scenario now in this world, that the immediate impact would be about $1 trillion. And that the annual cost for potential management of pandemic flu potential is at least $500 billion to $1 trillion a year just in anticipated outcomes. And so you would think if you're really, if you can translate those kind of dollars and say, well, let's spend $1 billion a year or $2 billion a year and develop a manufacturing capacity to do this work. It doesn't seem like a very difficult argument. That's like one airplane per year.
Well, I'd love to discuss this. There is a plan, for instance, for Americas, right? United States has a production unit, and Brazil has a huge production unit. And so if it was for just one virus, we can easily do in 4 months something like 200 million doses in the Brazilian facility here. So this would cover a good part of Latin America. And in 1 year, would cover completely Latin America. And the United States has about the same thing for all North America. And that's why some people from the U.S. Congress sometimes come down to Brazil to see how this facility is going, because there is a thinking about future or a vision that what are the threats like influenza. This is not shared by most of the other countries, unfortunately.
Yeah, I think the solution for manufacturing probably is going to have to come from places like Brazil, China, South Africa, Australia. We need to get the manufacturing out of Western society, because that's where we might have a chance of doing this at a reasonable cost level.
Other questions? I don't know. Did anybody send to the app? Because I didn't find it. Apparently there is nothing. Any other question? Adrian.
I just want Barney to comment on something. Barney, do you want to comment on what's the timeline for RSV getting on a shelf in a pharmacy?
Well, the good thing about RSV, I guess it's good, is that there is commercial value. The drug companies are very excited about RSV because, you know, this would be something for everyone who's old and every birth, which is about 120 million a year in the world. And so there's a big market for RSV. And now that we have evidence that this new molecule is immunogenic in a different way, there's two big pharmas who have the same molecule and who are competing with each other to see who can do it fastest. So they both did 1,200-person phase 1s last year. One of them is going to be reported at the end of this month. And one of them is going to be in pregnant women by the end of the summer. And so they're on very rapid competitive timeline for RSV. But the problem there is it has a market. What about these things that are more sporadic and maybe not generalizable? You need solutions for those things that don't have built-in markets. So RSV might, if everything goes smoothly, maybe licensure by 2023 or 2025, if it's smooth, which it never is.
So thank you. I think it's time to close the session. I'd like to thank Barney again.